A virus. A virus is any tiny, non-living particle they reproduce inside living cells. Since viruses are non-living, two of the characteristics of non-living things is that they do not have cells and they cannot reproduce by themselves. They're not capable of these, so they're considered non-living. Some examples of a virus is the flu, called influenza, chicken pox, the common cold, human papillomavirus, which causes warts, that's HPV that you hear about, and AIDS. AIDS attacks the immune system, so it attacks your white blood cells, which means you have no immune system. AIDS itself actually does not kill you. What kills you is all the other things that you contract because you do not have an immune system and you can't fight it off, like the common cold or a bacterial infection and things like that. So most viruses are considered parasites. The reason is because they do have to have a host cell in order to reproduce, in order to function. They have to be able to live inside and function inside of other cells. These are parasites if they have a host. Now, a virus's host is a cell individually, whether it's a regular cell like ours or a bacterial cell, anything like that. So they're very, very small. So whenever you look over here, this is malaria. It is a virus parasite, a viral parasite that actually attacks cells. So whenever you look, these are red blood cells right over here. And then this structure, this little tiny circular structure right here, this is the virus. Now whenever you look at this, this cell looks drastically different from this cell down here. That's because the virus will actually attach to the cell, inject its DNA in, make a bunch of these little particles that you see everywhere, all these little viral particles, and then it'll burst out and go to other cells and do the same thing. So almost all viruses are parasites because they will actually go in and destroy the cells in which they multiply. So whatever cell that they multiply in, they destroy it fully. Now there's a few distinct differences between a bacterial infection and a viral infection. And they're very easy to remember. You just have to use your common sense. A viral infection is going to be caused by a virus. Here are your examples. A bacterial infection is going to be caused by bacteria, such as these. Now, one thing unique about a viral infection, because it's a virus, it will not be affected by antibiotics. The only thing antibiotics can help us with is a bacterial infection. So bacteria can be killed by antibiotic drugs, while viral infections cannot. This is the transmission right here. Viruses are airborne, meaning we breathe them in. So most viruses affect our respiratory tissue. Bacteria, on the other hand, have to gain access, which means they have to go through our body, through water, cuts, infections, things like that. But they actually have to gain access. Now, since viruses are not living, they will die without a host, which are the cells that they reproduce within. Bacteria are living, So they can reproduce by dividing themselves. They do not require something else to be able to reproduce. The only thing that we have so far that can fight against viruses with us is what we call an antiviral. That is like the vaccines that we were looking at earlier, and those help combat viruses. What they do is they help protect the cells to keep them from being able to inject their DNA into the cell in order for it to reproduce. Viruses lack certain organelles which cause them to not be considered a cell. They lack the cell membrane, the basic outer structure. They have instead a bubble of fat that envelopes around the head. This is the head. This is the same exact structure as right here. It's called a capsid. It stays like this until it's ready to attack, and then it removes the the bubble of fat, exposes the capsid, and is able to then reattach to the cell. They also lack a nucleus because they just have their DNA inside of what we call the capsid, which is kind of like the nucleus of it, but it's not an actual nucleus. Now, whenever a virus wants to actually attach to its host, it has a tail and it has pins. Pins are the bottom part. That's what helps inject DNA. It helps insert the DNA. The tail are the legs that help attach it.
You ever seen anything like this before?
No. We're isolating the sick.
The truth about this virus is being kept from the world.
We just need to make sure that nobody knows until everybody knows.
Your wife was dead.
What happened to him?
It gets on airplanes. It travels at 500 miles an hour.
1 billion sick. That's where we're headed.
It's figuring us out faster than we're figuring it out.
Don't talk to anyone. Don't touch anyone. Stay away from other people.
Contagion, rated PG-13. In theaters and IMAX September 9th.
So, whenever we're looking at viruses, we need to understand how they reproduce inside of a living cell. So what you see here, this is a picture of an actual viral particle attached to a cell. Right here is the membrane of the cell. This is your virus. And now you see you have your legs right there, and you have your pin down here. So this is the pin, this is the tail. Now viruses inject their DNA into what they call their host cell, which is what we have as a parasite, or why they are called parasites. The virus forces the host cell to replicate its DNA, the viral DNA. Host cells are damaged and destroyed in the process when they're making new viruses. The viruses then release into the open to infect nearby cells. So the first thing that we have, the way a virus generally invades, is that you have a virus and you see it has this little fat layer around it to protect it. The cell will think it is something else because of that fat layer, and so it will engulf it, it'll eat it. Once it's inside of there, it gets rid of that fat layer and releases its DNA sometimes. Once its DNA is inside of the cell, it starts reproducing multiple DNAs for that virus. These viral particles then consume and take over the cell and burst out to go to new cells. Now you see the tails were stuck. That's the pin. It's attaching. It's injecting its DNA. It's coding for all the different parts of the cell of the virus particle, makes a bunch of different viruses, and then they break out. It's called lysis when they burst out like that. Now, this is really cool. This is what a virus looks like under a microscope. You can see here's its head and here is the DNA. This is its body, and down here are the legs, the tails. Now, there are two types of viral reproduction, just like there are two types of bacterial reproduction. Now, these two types are a little different. We have active and we have latent. Active reproduction is whenever, this is the definition for it, whenever they attach, whenever viruses attach and immediately begin to reproduce. When the virus attaches and begins to reproduce right that instant, that is active reproduction. So whenever you're looking at your picture, you have an active virus attaches, it's injecting its DNA into the cell. Now this is a bacterial cell, so this is one chromosome, because this right here is a bacteria. It injects its DNA into the bacterial cell. The virus's genetic material takes over the cell functions and the bacteria itself. You can no longer see the bacterial's DNA. Now, once all of this is done, they have all these parts, they get put together to make a bunch of little viruses, and the viruses will break out and go to nearby cells to repeat this process again. Now remember, that's active, so it's right now. Now we also have what we call hidden virus reproduction, which is the same thing as latent virus reproduction. Latent is the same exact process, except it occurs later than immediately. So let's look at the difference. We have a virus attaches to the surface of the bacterium. And now remember, it's using tails and pins to do this. It injects its DNA. Now this is the part that's different, right here and right here. These are your key parts. So now, instead of just immediately going over here, that doesn't happen. It goes down to number 3, where the actual viral DNA becomes a part of the bacteria's DNA. And then time passes. And we go up here. And eventually they wait for the perfect time to strike, pretty much. So they wait for the time to strike, and they wait until you're already sick, or it's that time of the year that your immune system is down, and then the viral DNA will remove itself and then become active again. So then we're able to go from step 4 to step 5 and step 6, where we are once it's now active, because see, it occurred later. It's now active. It makes all these little particles. They get put back together. They lysis out, remember? Lysis means they're breaking out, and then they go and repeat this cycle again. Now, if you look at this picture over here, you don't have this picture, but if you look at it, it's really pretty cool. This is a picture of what a bacterial cell looks like when viruses are trying to attach to it out here. And it's already having viruses, so they're breaking out. And they occur in mass numbers like this. Now, what we're going to look at with this video again, I want to show you how a virus is actually able and capable of tricking the cell into thinking it's not harmful. So whenever we watch this video, watch, and I'll show you the part that you need to really pay attention to. So whenever we're looking at this, the little green structures are the viruses. And right now the purple and pink are the viruses that are invading the cell. And now you see they have that fat coating around them. Which is why they're spherical, they're circular. Now that's a cell undergoing lysis, which means it's just bursting and exposing and releasing all of the different viral particles. And that's how small they are compared to normal cells as well. So whenever you look at this, you have your head, your body, your legs. Now watch. Right here. You see these are your tails. They're attaching to the cell. When they attach, smaller tail fibers come out, get inserted into the cell, and you see it squeezing down. What it does whenever it squeezes down, it releases a chemical right here into the cell that tricks it into thinking that it's okay, that it's not harmful. And so then it can further go in and inject its DNA. That's how it gets around the prevention of the cell fixing the DNA before it's able to reproduce. Now, whenever we're watching this, we have the idea, if you remember going back, that viruses are actually airborne. You see that? They're airborne. So whenever we have an airborne virus, which is how most of them are transferred, they affect nose, throat, and upper airways, which is in your respiratory system. So most viruses that you get, you have to inhale. They're carried through the air and they are breathed in, which is what inhale means. You breathe them in. And they infect the respiratory tissue so that you have sore throats, sinus problems, bronchial problems, you have the flu, all of that. The big thing is that viruses, you get inhaled and it affects your respiratory tissue. Some viruses now can affect the nervous system, like the West Nile virus, rabies, and encephalitis. This is what we were looking at in that video of Monsters Inside Me of the lady that had the parasite in her brain. The swelling was encephalitis. Now, rabies affects the nervous tissue of an animal and humans as well. And the West Nile, same thing. These are the three main ones that affect the nervous system. Now, we are able to immunize against viral infections because you know how you can take the flu vaccine and things like that. Well, that's what we're looking at, being immunized against them. So when a virus enters a body, it triggers the body's immune system, which are the white blood cells. When it enters it and it triggers that immune system, these white blood cells learn to attack and destroy the virus. If you survive, the white blood cells will remember the invader. Here's your key part. They'll actually remember that virus and respond more strongly to another attack the next time you come in contact with the virus. This is called natural immunity, whenever your body does this naturally, which is why a lot of people only get chickenpox once, because they come in contact with it once, they get sick, they come in contact with it a second time, and they're able to naturally immunize themselves against it. Now, we can also have a vaccine. Now, remember, a vaccine is where you have the dead or damaged particles. So these aren't dead viruses because viruses are not living, but they are damaged. They're broken into little pieces. So whenever you have your virus here, and its little DNA, its body, its legs, all of that, they take it and they just break it up into all kinds of pieces. And that's what gets injected into your body whenever you get the flu vaccine.
Its most dreaded disease is Ebola. There's a deadly outbreak in Africa. Experts from the U.S. Centers for Disease Control have rushed in, and ABC's Dr. Richard Besser, a former head of the CDC, is the only journalist with that team in the hot zone. He reports from Uganda tonight.
It's an 8,000-mile race into a hot zone. Dressed in a head-to-toe protective suit, I travel with an elite team of disease hunters from the CDC, joining a hunt for one of the world's deadliest diseases. Movies such as Contagion have tried to capture the terror of containing a virus. What we found is much more frightening. We make our way to the hospital, where Ebola patients are being treated. We are suited up in even more protective gear.
As high as it'll go without.
Every part of my body is covered, even a ski mask over my eyes.
There's not one speck of my body that is exposed.
One careless mistake can be deadly.
I have to say that there are a few things that I've done in medicine that are as nerve-wracking as going into this place.
We enter.
Those are the three who have Ebola. Over there. Over there.
and over there.
They don't know how these patients got Ebola, but we do know that very close contact with body fluids can spread it. Anytime someone touches something, they are quickly sprayed down with disinfectant.
Thank you.
What makes Ebola so terrifying, in as little as 2 days, there can be fever. Then, these patients' organs can fail. Uncontrollable internal bleeding can start. There is no treatment. More than half of those who get sick will die. Looking at the patients, the care is basic at best, and it is blisteringly hot. You can only stay in the suit for about 40 minutes. Doctors Without Borders leaves the effort on the ground here to make treatment more effective, and the CDC to stop the spread and figure out what caused it. CDC's Barbara Knust sees it as a critical mission here and across the globe.
We don't have high walls around our country. It's just because something happens far away in Africa doesn't mean that it can't happen back in the United States.
The feeling I had in that ward was absolutely frightening. That's not something we ever want to experience in America. That's why it's so important the CDC is there to help and to learn.
All right, so every time they said CDC, they're talking about the Center of Disease Control, and those are the people who produce our vaccines and antibiotics and antivirals and things like that.